let's talk about extranuclear inheritance. Again, with our 4 rules of Mendelian inheritance, here, typically in the 4 rules of Mendelian inheritance, this deals with nuclear DNA. So that's what the DNA that's present in the nucleus. So we're talking about the chromosomes. But that's not all the DNA that you get. So extranuclear inheritance, which is also known as cytoplasmic inheritance, because that's how these different organelles are inherited, refers to that inheritance patterns that involve genetic material outside the nucleus, hence extranuclear. The 2 most important examples are due to the genetic material that's present in these 2 organelles, mitochondria and chloroplast. Now, I'm not going to focus so much on chloroplast. What I am going to do is use some examples of mitochondria. Now, these are all found in the cytoplasm, and typically, when you talk about mitosis and meiosis, we rarely, if ever, talk about cytoplasmic inheritance of organelles. But there is a whole detailed process whereby these organelles are actually passed to those cells. So mitosis and meiosis deals with nuclear division. All right, so this is actually dealing with extranuclear division, and specifically in the form of organelles. Now let's review mostly what mitochondria is. Remember that mitochondria is, their main function is oxidative phosphorylation, basically the production of ATP. So this process is going to generate ATP. Oxygen is being consumed during the ATP synthesis, and then that ATP is used as an energy source to drive all cellular functions, not just things that are happening within the mitochondria. These are our power packs of the cell. Now, the genetic material in mitochondria is referred to as mtDNA or mitochondrial DNA. Human mitochondrial DNA consists of only about 17,000 base pairs. So in relation to nuclear DNA, there's relatively few genes in there. There's still quite a few, and they're critical. They're mostly ribosomal RNA and transfer RNA genes, but there's also 13 really important polypeptides that are encoded by the mitochondrial DNA that function in oxidative phosphorylation. So most of the other mitochondrial proteins are encoded by genes in the nucleus. However, these that really function in that oxidative phosphorylation are made in the mitochondria. The ones that are encoded in the nucleus are then passed into the cytoplasm and then transported into the mitochondria, which is a whole cool process that you will hopefully learn about in your cell and molecular or upper level classes. So these are necessary for synthesis of the proteins inside of mitochondria. So we have the mitochondria has its own ribosomes, and then there's specific transfer RNA genes for these amino acids in the mitochondria. These are the critical proteins that are involved in oxidative phosphorylation. And those of you who are taking biochemistry may actually recognize these genes. So the pattern of inheritance of these organelles is the pattern depends on what type of organism you are. In mammals, the mitochondria are inherited maternally or via maternal inheritance. So whether you're male or female, you get the mitochondria that you have from your mother. Now we can contrast that to the gymnosperms, where usually there's paternal inheritance of both the mitochondria and the chloroplast. In some organisms, you get some biparental inheritance, and that's seen in our yeast, our model organisms, and actually, think about, in bivalve mollusks, they actually exhibit doubly uniparental inheritance. So you can get it from both the mother or get it from the father too. So let's talk about this in terms of what kind of patterns we see in general. Typically in heterogamous species, this is where you produce two different types of gametes. You've got the female gamete that's very large, so that typically has all the organelles in it, whereas the male gamete is very small. It typically just carries DNA, and then other proteins that are needed for the movement of that sperm to actually access the female gamete. So in these species, the typical pattern is inheritance from the mother, or maternal inheritance. Now you can get some paternal leakage, but it tends to be relatively rare. This results in a phenomenon known as heteroplasmy, and can have some consequences depending on the type of mitochondria you're getting in terms of the paternal leakage. So occasionally you can get the mitochondria from the sperm. As I mentioned previously, the bivalve mollusks are a classic example of this doubly uniparental inheritance, in the sense that there you can have different kind of selective pressures on mitochondrial DNA from males and females, and that can result in large divergences between the two genomes on the scale of like the differences that you would see between two species. You could see that in the mitochondria of whether it comes from the females or the males in these bivalve mollusks. In the mouse, they've documented paternal leakage, and they actually see that for 1 to 4 paternal mitochondria are inherited for every 100,000 maternal mitochondria per generation of offspring. Now, the reason I'm talking about this is because it can have a huge impact on individuals in terms of human mitochondrial diseases. Since you are getting you have a strict maternal inheritance pattern, whatever kind of mutations are present in the mitochondria that you inherit from your mother is going to have a dramatic impact on you. Mitochondrial DNA tend to have very limited repair abilities, which is different from nuclear DNA. And the mitochondria are very susceptible to DNA damage because of the presence of ROS molecules, such as hydrogen peroxide. So these are reactive oxygen species, and they can attack the DNA and actually cause mutations of the mitochondrial DNA. Now, these kind of mitochondrial mutations occur in somatic cells, and they tend to accumulate, as all kinds of mutations do. However, if you start out with a mitochondrial DNA that's already mutated, then the likelihood of survivability of these embryos is very limited based on the type of mutation. If you have a mutation in any, like the NADH gene or the cytochrome oxidase genes, remember those are proteins that are going to be involved in oxidative phosphorylation. If you can't produce ATP efficiently or effectively, you're not going to make it. Okay, so there's been a lot of really cool work with introducing non-mutant forms of mitochondrial DNA into germ cells so that individuals can actually have children.